and welcome to your Chapter 3 lecture video. This is going to be our cytology lecture, so we're getting into the study of cells. So just to start off, these slides do have a lot of text. I'm going to try to not read all of it and just give you the highlights and explain some of the bigger concepts. So do make sure that you go back through and you read all of these slides and you read the chapter in our book as well. So a cell is the smallest unit of life when we're talking about how the world is organized and how things are organized. Yes, there are smaller things in a cell. There are macromolecules, there are atoms, there are subatomic particles, but a cell is the smallest unit of life. It's the smallest unit of organization that is able to exhibit the characteristics of life. It's continuously carrying out metabolic activities, meaning both building in creating new molecules as well as breaking some down, ingesting resources to then break it down through those catabolic and anabolic actions. It interacts to form tissues, organs, and organ systems. Again, thinking about how life and how structures are organized, cells do come together to form larger structures. There are about 30 trillion cells. We have a whole lot of them. They have different sizes, different shapes, and we'll see that they share some characteristics and then some are very unique to those areas that they serve. This allows them to have a lot of different functions and arrangements. The genes within a cell control a cell's action and responses. Every cell has all of the same DNA, but there's different areas of that DNA called genes that are going to be particularly active in a liver cell versus a kidney cell versus a skin cell. Here you can just see how they vary by shape. You have these elongated neurons for sending signals, these flattened epithelial cells, and these are muscle cells here. A composite cell is your general kind of cell. It is going to have a lot of the same common structures, but as your notes say, no cell contains all of these structures. It's going to be dependent upon what that specific cell's function is. Three things all cells are going to have, actually let me pull that back. Three things most of our cells are going to have are nucleus, cytoplasm, and a cell membrane. Your nucleus is going to house your DNA. That's the genetic material, the code directing the cell's functions. The cytoplasm, that is both the organelles, the structures, as we call them, the tiny organs, and the fluid inside of a cell. So if we were talking about the cytosol, that would just be the fluid, but the cytoplasm is the fluid and the organelles. And then the cell membrane, that's the boundary that covers the cell. These are eukaryotic cells. Animal cells do not have a cell wall like a plant cell would. They have the cell membrane that allows movement in and out of the cell. And as I said these things earlier, organelles are your little organs, just tiny, small structures. They used to, billions of years ago, just kind of all be floating around in a little pond together, and then eventually they were grouped together because they realized that they could be much more effective if their structure, if they were all kind of linked into one cell, that cell membrane being formed to allow them to house their functions together. And everything is dispersed in this fluid, that cytosol fluid. Here's an image of just a generalized cell. We're going to get into what all these structures look like, both here in lecture. We'll get into their functions. In lab, we'll definitely get into identifying these different features. But you can see that they have kind of projections on the outside, on the inside, all centered around this nucleus. So the cell membrane, that's also called your plasma membrane. Those are the same things. It is the semi-permeable boundary containing the cell contents. It regulates movement in and out of the cell. It aids in communication between other cells within the body, thinking about white blood cells maybe, or communicating to neighboring cells about different functions that need to happen. There's some general characteristics of a cell membrane. It's very thin, flexible, and elastic, so it can move and shift with our bodies as pressure is applied or as fluid increases and decreases, and that pressure in those areas shift. The cell membrane is able to shift with it as well. It is selectively permeable, so again, regulating entry and exit of substances. Our cells are not these completely walled off. They have to have some movement of things because as our cells make hormones, make proteins, and metabolic wastes, they have to be able to get rid of those as well as bring in fresh nutrients. And then some complex surface features, different cilia and flagella to aid in absorption and movement. Different proteins that are linked, different glycoproteins that help with signaling, all kinds of things. So when you see a picture of a cell, it's usually maybe got a couple of features on the outside of it, but really the outside of a cell is much, much more decorated with these identifying structures, features than we would think. It's kind of like I always kind of imagine it like all those when in space there's supposed to be all these satellites and things that are actually orbiting Earth. We don't see them because they're so small and they just kind of get disappeared. But in reality, we're just kind of surrounded by all of these satellites constantly. It's kind of how a cell is. It's got all of these things on the surface of it. We have to think about those. So looking at the structure of the cell membrane, of this plasma membrane, one common component are these lipids. So it's mostly composed of lipids and proteins, as well as a couple of carbohydrates. Some carbohydrates that get embedded in there as well. The basic structure of it is this this lipid bilayer, this phospholipid bilayer that has fatty acid tails and water-soluble heads facing the surfaces. So we'll see a picture of it in just a moment, but kind of think like a lollipop. And so it's both facing, I'm going to jump ahead, here we go. So I'll go back to those slides and I'll go through those, but I want you to see the picture of this plasma membrane. So these yellow fatty acid tails, you can see how they're pointing towards the inside, and then the blue round phosphate heads are pointing on the outside. So the lipid fatty acid tails, those are hydrophobic. They do not want to be exposed to water, while the phospholipid heads, those are hydrophilic. And so they want to be exposed to water. So wherever you see the heads, that's pointing towards either the inside of the cell. We can see here this is the intracellular space, or the extracellular space. And then the fatty acid tails, they line up together because they're avoiding the water. But this allows us to have this a delineated structure. If you didn't have this hydrophobic center, the hydrophobic tails with the hydrophilic heads orienting themselves in this way naturally, then the cell membrane wouldn't function as it's supposed to. You can also see embedded all throughout it these channel proteins, identity markers. Those are glycoproteins, carbohydrate chains that are linked to these receptor proteins and things, all just identifying the cell. And then embedded within the cell membrane, you can see these little rings of cholesterol, and this helps to give it some sturdiness, some structure. Because if you think about, just a drop, if you put oil into water, so that's what these fatty acid tails are essentially. They're a lipid, so just like oil is also a lipid. If you put lipid into water, it can easily break apart. It can get moved around. It gets, constantly can be broken back up and emulsified. So by having cholesterol embedded into the cell membrane, it gives a little bit more structure and rigidity to it. Okay, here we go. So yeah, just kind of going back through the structure, starting with those lipids. We talked about the phospholipid heads, the fatty acid tails. The lipid-soluble molecules are able to pass through. So respiratory gases, steroid hormones, they can just pass right through that membrane. It's impermeable to water-soluble molecules, though, so water has to pass through in certain, water-soluble molecules, rather, have to pass through certain channel proteins. And then, like I said, embedded cholesterol helps to stabilize things. There are also a lot of proteins found in the cell membrane. They're classified by how they're embedded, or where they rest within the phospholipid bilayer. So you have integral, transmembrane, and peripheral proteins. Integral are going to span the width. They can protrude to one or both sides, so both inside and outside of the cell, but they are integrated within it. Transmembrane proteins are type of integral proteins, but they will protrude on both sides. So a transmembrane protein is an integral protein, but an integral protein doesn't necessarily mean that it's a transmembrane, because transmembrane has to protrude both to the intra and extracellular spaces. Peripheral proteins are associated with one side of the bilayer. They don't actually go and penetrate through that lipid bilayer portion. They're just going to kind of be sitting in that phospholipid space, in one side of the bilayer. They vary in shape and function. Some act as receptors or growth factors on the cell surface. They start signal transduction. When we get into hormones and how hormones interact with cells, we'll see the difference between a lipid and a water-soluble hormone, and how water-soluble kind of start the signal transduction. And some act as transport molecules, so they create these ion channels for things like sodium and potassium to move in and out of the cell. So other protein functions, some have carbohydrates attached. We call those glycoproteins for cell identification. Again, that helps with the immune system because the white blood cells will wander around and they like to monitor and make sure if a cell is either a native cell to the body, or if it's a foreign cell, or if it is a native cell, is it healthy or is it infected? And cells can adjust, can send those signals based on these protein markers. There are certain proteins that go into the cell and provide support called your cytoskeleton. We'll look at those. And then some called cellular adhesion molecules, these CAMs, that help to interact with other cells and other cell structures. And again, there's just that picture again. You can see some of these cytoskeleton things, the channel proteins, receptor proteins, all that. So good question, go through here and see, can you identify which one of these are peripheral, which ones are integral, which ones are transmembrane? Cytoplasm, as I said, is both the fluid and the organelles within a cell. So it has the fluid, the cytosol, as well as these different organelle structures. The organelles each perform a specific function for the cell within the cell. So every cell is going to have some organelles, but no cell is going to have every organelle, because depending on what that cell's particular function is, it doesn't make sense to waste energy making a lot of extra structures that it's not really going to be useful for its function. Something that a lot of cells have are ribosomes. These are tiny spherical structures composed of RNA versus DNA, so ribonucleic acid instead of deoxyribonucleic acid, and a protein. They provide structural support, enzyme activity for protein synthesis, so ribosomes are essential in protein synthesis. Not membranous, so that means that they are not bound within the cell. They can float around and move around. But they are sometimes bound to the rough endoplasmic reticulum. So they can kind of both be in the cytoplasm, floating, free-floating, or bound to those endoplasmic reticulum, but they're not within the membrane structure of the cell. So looking at those endoplasmic reticulum, these are flattened sacs. These are flattened sacs that sit in rows around the nucleus. They act like a transport network all throughout your cell. There are two types of endoplasmic reticulum, the rough and the smooth. Rough are called so because they have ribosomes, and so when you look at them under a microscope, it looks like they have little bumps all over them, and they function for protein synthesis. So rough ER, protein synthesis. It has those ribosomes, that's why you can tell it's protein synthesis. The smooth ER do not have ribosomes. They function for lipid synthesis, absorption of fats, and metabolism of drugs. So still some really big important functions. So rough ER with those ribosomes are going to be protein synthesis, whereas smooth ER is going to function for lipid synthesis, absorption of fats, and metabolism of drugs, which means breaking down those drugs. Here you can see just kind of a depiction of them, a drawing of them. There's the nucleus, and then surrounding that you have the smooth ER, where there's no bumps, and then the rough ER that has these bumps. And here you can see that those are the ribosomes all over it, and then this one has no ribosomes, so it's smooth. Okay, so some other organelle structures, things like vesicles. Vesicles are a very generic structure. You see them all over the body. This is probably one of those things that almost all cells have. And these are just sacs. Just think like a little Ziploc bag or something, a little Tupperware container. They store and transport substances within or between cells. So if your cell is producing a certain hormone or a certain protein or something, it can package it into a vesicle and send it both within the cell and outside of the cell. The Golgi apparatus. This is more kind of flattened membranous sacs, right? It's going to refine, package, and transport proteins that were formed in those rough ER. So proteins are big structures, and they're not always completely made perfectly the first time. They have to go through some refinement to really be functional. And so your rough ER are going to make your proteins. They send them to the Golgi apparatus, where it does some refining and packaging up, putting things in those vesicles type structures. And they get sent throughout the cell or outside of the cell. You can see vesicles formed in the endoplasmic reticulum, take them to the Golgi apparatus. They can add sugar molecules to the proteins that help to stabilize their structure and enable folding. If you remember, proteins are required to be in a certain folded structure to work. Proteins can't be in a linear structure. They have to have that folded conformation to be able to be active. When vesicles are taking things out of the cell, just go ahead and give you a word here, exocytosis. Exo, exiting, right, out of, and then cytosis, the cell. So you can see the Golgi apparatus. It looks very, very similar to the smooth endoplasmic reticulum. So really just to be able to tell between these two structures, especially if you're thinking about for lab, I would give you its function. I would tell you that this one is involved in transporting and refining proteins. If it was the smooth endoplasmic reticulum, I would talk about lipid synthesis or metabolism of drugs, something like that. Mitochondria, this is usually something that a lot of us remember. We kind of get it hammered home in school as we're coming along. So the mitochondria, they are aiding in producing ATP. They are, as we say, the powerhouse of the cell. They are the structures that provide chemical energy. They go through these series of reactions, breaking down molecules and harnessing that energy of breaking those bonds to then force to rather recreate and regenerate these ATP molecules. Very active cells like your muscle cells are going to have thousands of mitochondria. Cells in your kidneys and your liver, they're going to have a lot of mitochondria, have a large energy need. And mitochondria also have their own DNA and reproduce by dividing. So even within themselves, within a cell, your mitochondria are going to have their own independent structures still, independent processes. There's your mitochondria. It's kind of got this kidney bean shape to it. It's got these cristae within the middle of them, these internal membranes, where to help divide and compartmentalize all of those oxidation reactions of breaking things down and putting them back together. Lysosomes and peroxisomes, a lot of these are helpful. They have enzymes rather that help with keeping the cell clean, right? I think lysosomes, I think Lysol, and so they have enzymes that break down nutrients, ingested material, worn out cell parts, break down cholesterol, toxins, drugs. So really helping to just kind of clean and tidy up throughout the cell. And then peroxisomes have a different set of enzymes than lysosomes. They help break down fatty acids, hydrogen peroxide, and detoxification of alcohol. So these have somewhat similar functions, but they do it with different enzymes, trying to kind of keep the cell nice and tidy. So we talked about the cytoskeleton a little bit and looking at the protein structures of the cell membrane. So microfilaments and microtubules are little thread-like structures that serve as the cytoskeleton of the cell to help give it some structure. So cells are supposed to be malleable. They can flatten, they can squish, they can move, but they have these cytoskeleton network to help give it a general shape. So when it isn't under kind of deformation pressure, it can go back to that original shape. It also acts as a transport network, kind of like railways within the cell, so that as vesicles are being transported from one place to another within a cell, they have this pathway they can go along rather than just kind of bumbling and bumping through the cytoplasm. Microfilaments, they're made up of actin, which is a certain type of contractile protein, cause various cellular movements. Microtubules are a globular protein called tubulin. And they form this long spiral tube, important for cell division. We'll see those later in this lecture. They help with organization of materials as the cell is dividing. And intermediate fibers made of different proteins, abundant in skin cells and neurons, more so than other cells, and again are helping to provide that structure. Here you can kind of see a depiction of it. This is an electron microscopic image of the cytoskeleton structure within a cell. And then here are just some kind of drawings of it. You can see how they're kind of connecting to different structures to keep things in a general place. All right, centrosomes. Centrosomes, again, are going to be important for cellular division. They're these two hollow cylinders called centrioles that function in the distribution of chromosomes. They help to line up the chromosomes so that way they can be divided evenly. Cilia and flagella are both external structures on the cell. The cilia are abundant on free surfaces. They move in a wave. They kind of help to move stuff along the cell wall, in the cell membrane rather, whereas the flagella is going to help to actually move the cell. Usually you will see a lot of cilia. You see them along the respiratory tract. It's not the nose, the hairs in your nose. Those are a different kind of hair. Cilia are microscopic, and they help to make sure that mucus and other structures, dirt and things don't get caught on top of a cell. They get brushed along the cell surface and they don't kind of clump on top of it. Smokers tend to lose these cilia, which is why they get that smoker's cough. Their mucus tends to hang out more because it's not being washed away by the cilia, by these hairs kind of moving them along. The mucus tends to sit on the cells, and they cough it up more. So usually if you're going to have cilia, there's going to be a lot of cilia covering the cell, not involved with movement. Whereas flagella, there's one flagella on a cell, and it is involved in movement. I'm thinking like a sperm cell. Here you can see those centrosomes. They're kind of arranged around in the centrioles to then, sorry, it's the centrosomes are the individual things, and collectively it's called a centriole. And again, these are helping with cell division. These are cilia, again, just these hairs covering the cell, and then flagella in your sperm cells. All right, so the nucleus. The nucleus contains your genetic material, your DNA, deoxyribonucleic acid. This is what's going to have the coding for cell processes, proteins, that kind of stuff. You usually have a large nucleus surrounded by its own envelope, so it's kind of enclosed within itself, and nuclear pores that allow passage of certain substances, whether that's enzymes going in, ribosomes going out, proteins going out, things like that. Inside of your nucleus, you have your nucleoplasm. So inside of that nuclear envelope, rather, you have the nucleoplasm. So that's going to be the fluid and the structures. There's also what we call a nucleolus, and this is a dense body composed of RNA and protein, and this is where your ribosomes are produced. So within that nuclear envelope, within the that's surrounding the nucleus, you have the nucleolus as well, making RNA or making ribosomes. Ribosomes are then, remember, used for protein synthesis. The structure called chromatin is your DNA found in the nucleus. So depending on the state of the DNA, whether it is condensed down or loosely uncoiled, we call it something different. And depending upon the state of division that the cell is under, that's going to determine if it's chromatin nice and loose or if it's more condensed into chromosomes. So at the end of this chapter, once we get into the cell cycle, we'll kind of talk more about that, and we'll connect the DNA structure with the cell cycle structure. But just kind of suffice it to say that chromatin is very loosely uncoiled DNA, and it has to be loose and uncoiled because that is what allows it to be read and translated into proteins. When DNA is condensed into chromosomes, which normally when you see a chromosome on a karyotype, right, it's kind of in those X shapes, and you can kind of line them up and you match them up or whatever. That is its condensed form during cell division, because you want it to be very condensed so that way it can divide evenly. But again, it's not functional as a chromosome form because it's so coiled, the structures can't get in there to read it and translate it. So chromatin is this loosely coiled DNA that allows it to be read. Oh, wrong way. There we go. So in here you can see this chromatin, this kind of very loose, unspoiled, or uncondensed DNA. This entire thing is your nucleus. It has the nuclear envelope with nuclear pores, these little holes. And then the very center is the nucleolus. So that dark center in the middle is the nucleolus. And in this next couple of tables, they just kind of go through what I just said. It has the cell part, the structure, and then the function. You should definitely be reviewing these tables so that way, just as a way to help you know what you're looking at. And again, remembering the function even for lab, because this is what I'll give you. I'll tell you the function of something to help you identify what the structure is. All right, so now that we kind of talked about the basic structures of a cell and give you the idea of what the cell membrane is doing, let's talk about how things get into and out of the cell. So movements into and out of the cell. So our membrane is this selective barrier. It has mechanisms that allow things to move across. Those can be either passive, that don't require energy, or active, that do require energy. Going into or out of the cell, it really just depends on what the structure is, what is trying to pass through the cell. That will determine if it's passive or active. Some passive transport mechanisms, things like diffusion, facilitated diffusion, osmosis, and filtration. So don't let this word facilitated diffusion make you think that it requires activity. Facilitated just means that it needs a protein for it to happen. The rest of these can kind of happen just through the cell, the plasma membrane, without any protein support. So facilitated is still passive, it just requires a protein channel. And then active transport mechanisms are things like active transport, endocytosis, and exocytosis. We'll look at these two especially in more detail. So starting with diffusion, just your simple diffusion. So simple diffusion means that a structure can just pass through the plasma membrane. It does not require energy, and it does not require a channel protein for it to pass through. It can actually pass through the phospholipid bilayer without any other aid. So simple diffusion happens when you have movement of molecules or ions in a liquid or gas from a region of high concentration to low concentration. So this is moving down its concentration gradient. Let's say that you have a lot of glucose molecules outside of your cell and not very many inside of your cell. They can move passively down that gradient into the cell without needing any energy for it to happen. It's caused by random motion collision of particles because things don't want to be in a crowded spot. So if there's a whole bunch of things on one side of the cell membrane, they're going to naturally want to move across it down their concentration gradient, so there's not as much bumping and activity happening. At diffusional equilibrium, they have become uniformly distributed. They will continue to move, but it's an equal one-to-one change. It's not like you have more going one direction than you do the other. Diffusion enables oxygen and carbon dioxide to go between air and the blood and the lungs and between the blood and cells. So when you take a deep breath in, your lungs now have a lot more oxygen-rich air within them, and so that oxygen is going to naturally flow into your bloodstream, and then from your bloodstream, that carbon dioxide is going to naturally flow into your lungs, so that you can then exhale it out. And so that all happens based on diffusion. Diffusion only happens if the cell membrane is permeable to the substance and there is a concentration gradient. So those two qualifications have to be met. The cell membrane has to be permeable to it. A lot of times that has to do with things based like charge or size of the structure or the hydrophobicity of the structure. And then again, that idea that there is a concentration gradient. There has to be this unequal distribution. And thankfully the cell membrane is what allows for there to be a concentration gradient. Because if there was no cell membrane and you were just a big ball of liquid, there would be no difference between one side and another. But by having a cell membrane, by having this barrier, we can establish this concentration gradient. And we'll see, and especially in the active transport, how our cells utilize that concentration gradient to harness energy. So a classic example is a sugar cube in a glass of water. If you put the sugar cube in there, it will slowly start to dissolve. And then as it dissolves, you can see how the sugar molecules will kind of naturally disperse evenly throughout it. Even if you don't stir it, it will naturally happen. Obviously, if you stir it, if you add that agitation, it will distribute evenly more quickly, but it will still happen on its own. So diffusion now across a permeable membrane, so still have this kind of sugar water, but now we're adding this membrane that is permeable to both water and a solute. Okay, so water is obviously the solvent, right? So everything is being dissolved into water, and the solute is the solid. It's the thing that's being added into the solvent and creating the solution. So at first you can see the little blue ones are water molecules, the big red ones are the solute, it's what you're adding. So this could be your sugar molecule that you're adding in. So at first you put in your permeable membrane and you have equal distribution of, or rather you have more water molecules in the B side, fewer molecules in the A side, but you have all of these solute molecules here. So since this membrane is permeable to both water and the solute, they're each going to move down their concentration gradient. So from A, you're going to have a lot of these solute molecules that move over to the B side, and then from B, you're going to have all of these excess water molecules move over into the A side. And they're going to keep doing this, and as time passes, they eventually come into this equilibrium. Because this membrane was permeable to both of them, they're able to move across both and find that equilibrium, that balance. Sorry, just go back here real quick. What could happen is that this was only permeable to one of these structures. Then you would have this unequal distribution. Let's say, again, we're starting over here in A. If this was only permeable to water, you would eventually have water moving out of B and into A. So even though the volume would change, the concentration is what the molecules are trying to go for. So molecules don't think in terms of, oh, let's keep the volumes equal. They're thinking about, we have to get this equal distribution of concentration. And so if this was only permeable to water, you would just have B moving into the water from B moving into the A side and no movement from A. Here you're seeing that diffusion of O2 and CO2 in our blood. So this is our capillaries moving through our lungs, or rather, I'm sorry, this is at the tissue level. So this is a blood capillary delivering oxygen, fresh oxygen and nutrients to one of your body cells. So your blood and your capillaries is going to have a higher level of oxygen. Those molecules will jump out into your cell. And then because your cell uses oxygen, it has a lower amount of O2 and a higher amount of CO2 because that's part of a waste product. So that higher CO2 is going to naturally diffuse into the blood capillaries, which will turn into veins, which eventually connect to the lungs and allow us to exhale out. But this is, again, going from high O2, low CO2 in the blood, and low O2, CO2 in the cells, and then those go down their gradients. And this is also why we talked about the cell membrane being very, very thin. That allows for these molecules to pass through. Same thing with these capillaries. They're just that one single cell layer. Okay, so facilitated diffusion. We said passive diffusion, they don't really need any other structures to aid in it, but in facilitated diffusion, they use membrane proteins to carry the substance across the phospholipid bilayer. So maybe it's too large or something like that, and so it can't pass on its own, and so these integral proteins help with that transition. One type involves the use of ion channels to transport ions across the cell membrane. An ion is it's a charged molecule, and so a lot of times charged molecules can't pass through the plasma membrane because their charges will be repelled. Substances like glucose, amino acids, they can't pass through the phospholipid bilayer since they're water soluble or too large. Glucose especially, it's water soluble, so it can't just naturally pass through those lipid tails. So it has a carrier molecule that aids with that. The number of carrier molecules in the cell membrane limits the rate of this process. So with something like simple diffusion, it can happen as fast as it needs to, as long as it needs to, because it's just those molecules moving on their own. But when it comes to things like facilitated diffusion, that is rate dependent upon the number of carrier molecules. And so if your cell only has, a certain, is only going to be limited by how many it can do at a time. Kind of thinking about like if you're at the airport and you're trying to get your luggage and there's only a few people that can work the luggage carousel, well, they can only move things through so fast. Or maybe like the last time I went and they had 2 gate agents working the entire security TSA check. It's like, okay, we can only move as fast as those 2 people can go. And so that's that rate dependent process. But again, this is still facilitated diffusion. It's not active, so it's not dependent upon energy. Here you can see an example of this protein channel, this ion channel rather. And so, no, I'm sorry. This one is these are protein channels, and this one is an ion channel. This is the outside of the cell, the extracellular space versus the intracellular space. Intracellular is going to have this lower concentration of these ions and proteins, and so these structures still naturally want to move down their concentration gradient. So they still want to come into the cell, but because of their charge, because of their size, they can't fit through this membrane, and so they use these ion channels and carrier proteins to allow them to go through. And it's very interesting, these structures, so let's say this is a protein right here, it's going to bind to this carrier protein, and when it binds, it causes the carrier protein to actually change structure. So it actually changes its shape, and by changing its shape, it turns into this one here, where now it's open on the inside of the cell versus the outside of the cell. And when it opens on the inside of the cell, it pops the protein out. And then what happens, because now this protein is no longer bound in here, it switches back to this original shape where it's open and ready to bind another protein. And so the protein binding changes the shape. The change of the shape kicks the protein out. the protein kicks out, it goes back to its original shape. And so it can just happen pretty quickly. But again, you have to rely on the number of these proteins. You have all these proteins waiting for these channel carriers. Same thing happens with the ion channels too. Okay, so now looking, still passive process, but this is osmosis. Osmosis is diffusion, but exclusively for water. That's essentially how you can think about it. Osmosis is exclusively referring to the movement of water across a membrane. It's a special case of diffusion. Water moves across a selectively permeable membrane, still going from greater to lower concentration. So yes, let's say your cells are dehydrated, they don't have as much water as they need to, so water will naturally move from your bloodstream into your cells. And then you'll start to feel thirsty, and so you'll drink more water, and that water will flow into your bloodstream. And then if you're still dehydrated at your cellular level, that water will then seep back into the cells until eventually you're hydrated enough and enough water has moved into your cells that they're balanced. During osmosis, water moves from a region of lower solute to a higher solute concentration. So this is kind of the other way of saying that. So I can talk about how water moves from a place of a high water concentration to low water concentration. But when you have a lot of water in one area, that is naturally going to dilute the solute concentration. Imagine if you had kind of a glass of sugar water, you could add more water to that glass, and it would taste less sweet, but there's still the same number of sugar molecules, but there's more water in there now. If what happens is that water will move not just from an area of higher water concentration, but it will move to an area that has more solutes. So if you have two containers that are connected to each other, and one has a lot of sugar water in it, and the other one only has pure water in it, that pure water is going to move to that sugar water area to try and balance out the solute concentration, to try to make it the molarity more equal. Just want to make sure I said everything from this slide. And then that creates this osmotic pressure. So osmotic pressure is the pressure needed to lift a volume of water. Greater concentration of solutes in a solution, the greater its osmotic pressure. So essentially with the movement of water, think about like a river or a waterfall or something like that. As water moves, it has this pressure, it has this force behind it. And so how much of that force is going to be required, that pressure is going to be required to move that volume of water. And so the greater concentration of solutes in a solution, so the more hypertonic something is, the higher your osmotic pressure is going to be. So here you can see this glass. We kind of talked about this before. You have a selectively permeable membrane now, and so it's only going to let certain things pass. And as you see on side A, you have more of these proteins. Side B has more water. What's going to happen is that the water will leave side B and go into side A to try and balance this solute concentration. And if you were to go in here and count the water molecules versus the protein molecules on A and B, and then count them here, A and B, you would find that in this one they're an equal ratio. So even though they're unequal in actual number, the ratio of water to protein molecules is the same on A as it is on B. Okay, so then I used this word just a moment ago, the tonicity. So tonicity refers to the composition of dissolved solutes in a solution surrounding a cell. So the composition of dissolved solutes in a solution surrounding a cell. So now we're not just talking about inside of the cell or the plasma membrane. We're talking about that area outside of our cells, just that extracellular space. So that's where a lot of things tend to float around and kind of move around as they're going into the cells or into our bloodstream. They're going to have to float in this in-between space first. And once their concentration starts to get high enough, then they start to move back into the cells or into the cells. That's going to be one of those driving factors. So tonicity affects the size and shape of a cell. The greater the impermeable solute concentration, the greater its osmotic pressure. So again, the more solutes you have in an area, the more force it's going to push onto from water that it's going to be requiring. Water moves towards solutions with greater osmotic pressure because that's the force, that's the direction that water is being pulled into. Cell membranes are usually permeable to water but impermeable to many solutes. So water equilibrates through osmosis, equilibrating the osmotic pressure in the intracellular and extracellular fluids. So what this sentence says is that water can move pretty freely in between through the plasma membrane, but the solutes can't. And so rather than having a bunch of solutes being moved back and forth, you have a lot of water that gets moved back and forth to balance out the osmotic pressure and the concentration gradients on the two sides of the cell. Where this comes into play clinically is very important, because it's one of the reasons why when you're giving an IV, you're not just giving pure water, you're giving a saline solution, because a saline solution has a little bit of salt in it. Because when we're talking about solutes, I keep talking about solutes. I should go ahead and give you some actual structures. A lot of times solutes tend to be sodium, chloride, magnesium, and potassium. Those tend to be a lot of the solutes that we're talking about are salts. And so when you're giving someone an IV fluids, you're giving them saline solution, and it's typically this 0.9% NaCl, sodium chloride solution, because it is isotonic to human cells, meaning that this 0.9% saline solution is going to be very balanced. So it will allow water to equally and evenly move between the bloodstream, the extracellular space, and inside of the cells without causing these disruptions in cell shape and cell size and causing these big pressure changes. If you were to, let's say, give a hypertonic solution, this would be like giving somebody really, really salty water. You're like, yes, you're giving them water, but you're also giving them so much solute and so much salt that that's going to cause water to exit their cells to try and dilute the salt that's in your bloodstream, because that salt won't move quite as fast as water will. And then the opposite is low osmotic pressure, something that is hypotonic. And this is why you can, the hypotonic one is one of the reasons why you can actually die from overhydrating yourself. I see it all the time that, people go to the gym and they're carrying around their big gallon jug of water. And you have to remember that when you go to the gym and you're sweating, you're losing water and you're losing salt. You're losing those solutes. And if all you do is replace the water, you're just drinking pure water, this is what happens, is that you start to get this hypotonicity in your blood, to the point that your water won't enter your cells. Your cells will actually, your water will enter your cells and they'll actually start to explode and they can rupture. Because they have so much water moving into them, because it's not an equal balanced isotonic solution. So whenever you're giving injections of something, it's almost always done in an isotonic solution, and we watch out for these hypotonic areas. The way that I remember it is that hypotonic, you have H2O. Right, hypo, H2O. And so hypotonic solutions have a lot of water. Hypertonic have a lot of solutes. They don't have very much water. That's just kind of the way that my brain works. Hypertonic solutions are going to dehydrate your body, right? The cells placed in a hypertonic solution lose water and shrink. If you are drinking pure salt water, like let's say you're lost at sea and you start drinking ocean water, salt water, to at least try to stay hydrated, your cells will actually start to get even more dehydrated because the water will leach out into your bloodstream trying to dilute the salt. Versus hypotonic, like I said, it's where you are maybe sweating a whole lot. I worked in horse farms when I was growing up, and so we always tried to make sure we were drinking Gatorade or making sure you're eating as you're hydrating, so that way you have those solutes and you're not just drinking pure water, because that can, again, kill you just as fast as hypertonic cells will, and possibly even a little bit faster. Here you can see what happens to our red blood cells when they're placed in different types of solutions. So a red blood cell in an isotonic solution, it has an equal movement of solutes and water, so all is good. In a hypertonic solution, you have a lot of salt in this in your water here, in your solution outside of the cell, and so water is going to be moving out of your red blood cell into the solution to try and balance it. And then in a hypotonic solution, you can see you have a lot of just pure water, and a cell being sat in that water is going to move into your red blood cell to try and balance those solute solutions, and that causes it to rupture. Okay, some more passive mechanisms then. Filtration. Filtration is the process of forcing molecules through membranes due to the exertion of pressure. This is a lot of times what happens, like in our kidneys. They're going through filtration. You have in lymph nodes as well. You have fluid that's being pushed through at a high pressure, and so that causes water to get filtered out, or structures or stuff to get filtered out. Let's just say stuff. Blood pressure comes from the action of the heart, which is a type of hydrostatic pressure. So blood pressure is a type of hydrostatic pressure. It's that fluid pressure, and that's the force of filtration all throughout your body. It's used to produce tissue fluid from blood plasma, water in small solutes, sent through the walls of capillaries to deliver oxygen, nutrients to the cells, while large particles like plasma proteins remain in the capillaries. So essentially, what you have is in these capillaries, the force of your heart pumping doesn't just push blood along the vessels, it also at the capillary level pushes fluid and small structures out of the capillary so they can go into the cells, but it's not so high of a pressure that it's pushing proteins and things like that out. Those exit in different ways. They have different mechanisms for exiting into the cells. But filtration pressure from your heart is what allows just the delivery of small O2 and nutrients and things like that. Particles generally limited in movement by the size in relation to the opening of the walls of the capillary. Again, this is why things like high blood pressure, if you have high blood pressure and elevated heart rate or things like that, you're putting extra strain at the capillary level, and so you're potentially filtering extra stuff. You're filtering out proteins, which should be staying in your bloodstream, but because the pressure is so high, those proteins kind of get clogged in the capillary pores, and they just get pushed out eventually, and then those openings are larger, and so you end up losing more fluid, more oxygen, more nutrients than you would normally. And so then your heart has to work even harder to maintain those processes and those proper pressures. So here you can see this is the blood vessel. It's got different structures. We're just going to call this oxygen because it's blue and small. And then you have these larger kind of molecules that aren't being allowed to exit. So each of these little overlaps here in between the cells of the capillary, this is where those small particles can exit. So as fluid is moving in this general direction, you can see the direction of blood flow. Blood pressure is forcing them out. It's forcing these small molecules out, but these large molecules are still staying in because the pressure isn't that high. Okay, so that was of our passive processes. Looking at some active stuff. Active transport, these move substances from a region of low concentration to high concentration. So this is opposite from going down its concentration gradient, it's going up its concentration gradient. And that is why they require ATP. They require energy and a carrier molecule. Okay, so just remembering that active mechanisms, they're going against their concentration gradient. They require energy and a carrier molecule. So active transport, process of moving substances from low to high concentration through carrier molecules using adenosine triphosphate. The carrier proteins can be called pumps. That's something that we talk about. So channels are a passive process, pumps are an active process because they're literally pumping things against their gradient. They talk about the sodium-potassium pump. As much as 40% of a cell's energy can be used to fuel this process. And some of the most common ones are sodium, potassium, calcium, and hydrogen, as well as some sugars and amino acids. Those are all used active transport. So here you can see the sodium-potassium pump. We have the intracellular space in here, the extracellular space out here. So this pump is always going to be exchanging 3 sodium for 2 potassium. That is just the rate at which it does. That's how this one works. Your cell has your cell has fewer sodium on the inside, more on the outside. And so you have these 3 sodium that are going to bind to this carrier protein, or to this pump. You have the 2 potassium that bind to the pump. And then because they're going against their gradient, because again, there's already a bunch of sodium out here, these 3 don't want to go outside, but because they're using energy, it allows them to be pushed out. So this ATP molecule binds, and as it binds, it causes this pump to essentially slightly change its shape, and it ejects these 3 sodium out and these 2 potassium in. And because you're constantly losing a little bit more positive than you're gaining, you're losing 3 positive ions and you're gaining 2 positive ions, for several reasons, but that being one of them. The inside of the cell is always a little more negative than the outside of the cell. And we'll see. There is, again, one, it's to keep the reason that our bodies are doing this, yes, is to keep this unequal charge, to help with that unequal charge. But then we'll see in other lectures where it uses the sodium, all these kind of sodium that are built up in here, and all these potassium built up in here. It uses that. It allows them to move down their concentration gradient at a certain point, so that way they can harness that energy as well, that passive energy. All right, and then endo and exocytosis. Endo is going inside, exo is going outside of the cell. These are typically used for large substances without crossing the cell membrane. So some substances are so large that if they did try to cross the cell membrane, they would end up bursting the cell open. And so by using these processes, they can fuse with the cell membrane, and then a portion of the cell membrane is pinched off and then just kind of naturally closes back and fuses back together without causing a major disruption. So endocytosis, molecules are too large to be transported by other means. They're conveyed into the cells inside of a vesicle that forms from a section of the cell membrane, whereas exocytosis is the same thing, except it's going outside of the cell. There are some specific types of endocytosis that we can look at, pinocytosis and phagocytosis. Pinocytosis is cell drinking, so it actually will just take in a large a molecule of water, a large vesicle of water can just be drawn in into the cell rather than just individual molecules diffusing through. Phagocytosis, this is the eating, the cell eating. So it's actually engulfing. And it's not that your cell needs to eat. Your cell gets nutrients through oxygen and glucose that are brought into it. But this is for like if you have a phagocytic white blood cell, a macrophage or something, and so it encounters a bacterium, and so it's going to kind of form this structure that reaches out, encloses the bacterium, and then pulls it back inside of itself. And that obviously, a bacterium can be a large structure compared to, at a cell size, compared to, a molecule of glucose or a protein or something. And so that would be too large to just allow it to move through the plasma membrane. It actually has to form this use its plasma membrane to form a vesicle around the bacterium and then pull it inside. Once inside, then it gets digested by lysosomes. And then receptor-mediated endocytosis, which the cell takes very specific molecules that bind specific receptors on the cell membrane to initiate vesicle formation. So you have these ligands, so these molecules that are kind of floating through your bloodstream, and they signal your cells by binding to specific receptors that then initiate this intake of whatever the structure might be, whatever the hormone or signal might be. So here you can see both your pinocytosis and your phagocytosis happening. You see how the cell kind of creates this little indent, and then these edges kind of move up and around the structure that's being engulfed, and then it pinches back together here, and then what happens is that the vesicle can be released inside of the cell, so now they have been pinocytized and phagocytized, but your cell membrane remained intact the whole time. Your cell membrane was never broken. Once it's inside, the lysosome molecule will detect this vesicle with the ingested material, whatever it may be, right? And so it forms this phagolysosome, and then these enzymes are released, and it starts to break them down. And then once it's broken down, anything that can be absorbed and used will be, and then anything that needs to be expelled can be excreted and expelled. And again, it does it through exocytosis. So exact same thing where it fuses to the inside of the plasma membrane, that starts to open up and those can be released. And then your plasma membrane just kind of incorporates those vesicle components. This is that receptor-mediated endocytosis. So you can see these ligands floating around here. They bind to these receptor proteins, and then that causes them to take them in the vesicle forms, enclosing and transporting receptor combinations. This might be like a lipid-soluble hormone. Will use this kind of pathway to enter the nucleus and cause protein synthesis. So definitely use these tables, go through the different mechanisms of passive and active transport. Be familiar with those. All right, now let's get into the cell cycle. So our cells are on this growth, this constant growth, development, and then apoptosis, the cell death cycle. Uh, the cell cycle is a series of changes that it undergoes from time to time, from the time it's formed until it divides. The main phases are interphase, mitosis, and cytokinesis. Daughter cells might then undergo changes to become specialized. I'm just reading through this real quick. So when we're talking about this version of cell cycle, we're talking about division of our non-sex cells. So this does not refer to sperm and egg cells. This is only referring to your somatic cells, things like your hair, your nails, your skin, your liver, your kidneys, your tongue, everything except for your egg and sperm cells. These cells go through mitosis. And so you can think about mitose, right? So these are the division cycle that your just regular body cells go through. They have these three phases. Interphase is their growth phase. So think about this like from childhood to puberty. So those first 12 to 15-ish years of your life, you're just kind of going about things. You're getting taller, you're getting bigger, you're eating, all those kind of things. And then eventually you hit puberty. And at puberty, that is, even though you are a child and we do not let children have children of their own, your body is ready for it. Your body is ready for that next stage of development. And so for our cells, what's happening is that they go through this interphase phase. They go through this interphase cycle, and then once they've kind of grown and developed enough, they're ready to divide, and that's when they go through their mitotic cycle, their mitosis. And you have one parent cell that gets divided into two daughter cells, two identical daughter cells. And those two daughter cells then go through the process of interphase again, and they grow, grow, grow until they're large enough, and they go through their mitosis. Cytokinesis is part of the division cycle. Mitosis is referring to this division of the internal components and the plasma membrane, the genetic components, and then cytokinesis is the actual division of the fluids and things in the cell separating. So they happen, these two, cytokinesis and mitosis happen concurrently, but we're just dividing them out here. And then you can read through this, talk about checkpoints and restriction checkpoints, because there's all these different signals that control how fast it happens, when it's happening, and what cells are going through the division cycle at what time. Most cells do not divide continually. They have a maximum number because of the telomeres on the tips of the chromosomes. With every division, they get a little bit shorter and a little bit shorter. And so eventually those cells have to be recycled and go through this programmed cell death. Because if they don't, that's when you start to get genetic mutations, and you can have things like cancerous cells that get formed and tumors getting formed. So here's just a depiction of the phases of a cell cycle. So we can start here at this interphase. So we break interphase up into three portions: the G1, S, and G2 phase. This stands for gap. So the gap 1 phase, the S phase, and the gap 2 phase. G1 phase is cell growth. So again, you have this kind of just period of time where the cell is just growing, getting larger, incorporating nutrients, all that kind of stuff. And then at a certain point, once it gets large enough, it signals into this S phase where you have the genetic material replicating. And that's when your DNA is going to replicate. Because if you have one parent cell making two daughter cells, those two daughter cells have to have the full complement of genetic material. So you can't just take the parent cell and divide the genetic material in half, because then those cells would be incomplete. So the parent cell is going to go through this S phase of replicating its genetic material. And then after it does that, you know what? It's tired. Haven't you ever just had one of those days where it's like, oh, I'm just exhausted, and and you didn't even, maybe it was like a day like I had where it's just like, oh, I just took my dog to the vet and I'm exhausted now. So after you've gone through this kind of S phase, there's this kind of resting phase that happens, this G2 phase, some more growth that's going to go on, and then it goes into mitosis. And you can see mitosis has 4 steps to it: prophase, metaphase, anaphase, and telophase. And then within anaphase, telophase, you have cytokinesis that happens as well, which is, again, that division of the cellular contents. Mitosis, these 4 are really talking about the division of this genetic material, because now your parent cell has twice the genetic material that it needs to, and so that has to be divided evenly. And so it's these four things, these four phases of mitosis that allow that to happen. And then once those two daughter cells are formed, they go through it. They go through their growth phase, S phase, G2 phase, and then mitosis. You can see, though, that this restriction checkpoint, they might be going through division. Some of them can go and be specialized, and then some are going to go through apoptosis. And so it just depends on what that cell is programmed for. Interphase is the phase of the cycle before cell division actually begins. The cell is growing and synthesizing new molecules, membranes, DNA, and organelles. A lot of synthetic activity, meaning creation of structures rather than the time of rest. The gap and growth phases, whatever you want to call those, that's when the cell grows. Structures other than DNA are duplicated. So your mitochondria, your centrioles, those kind of things. And the S phase, the synthesis phase, is the DNA cell replication. So as I was saying, there's two types of cell division. I think I just kind of talked about how we're focusing on mitosis for this one. The other one is meiosis. So meiosis is for sperm and egg cell production. Those are your gametes. Gamete is your sex cell, so your sperm and egg cells. And this just makes sure that those have half the number of normal chromosomes as your other body cells. And that's because, as I was saying, in mitosis, your parent cell forms two daughter cells, and those daughter cells are your normal body cells, so they need to have the full complement of genetic material. But with your gametes, so for males, their sperm, those are going to be joining with an egg, and so it's okay if a sperm and an egg both have half the genetic material, because when they come back together, they're going to have all 46 chromosomes, and that's why you are a blend of both your parents, because you have this combination. You got half your genetic material from each of them, and they get blended together and recombined when the sperm and egg rejoin. And so meiosis is basically mitosis, but it goes through twice. But it's, again, in meiosis, we look in chapter 19, chapter 20, I think we get into meiosis. But it's just 2 rounds of mitosis is basically what it is. With cell division, mitosis, and cytokinesis, again, this is the more common type, used to increase the number of cells for growth, development, healing. Mitosis is division of the nucleus, and cytokinesis is division of the cytoplasm. So they're regulated to happen kind of at the same time or in concurrence with each other. So that way you have an even division and you can have 2 healthy daughter cells produced. And those are going to have the exact copy of the original genetic material. When you go through meiosis, you get this mixture and blend of genetic material. But your daughter cells should have the exact same copy as your parent cells. So mitosis happens in a series of 4 stages, but it is kind of a continuous thing, so one overlaps with the other. The first is prophase. DNA condenses into visible chromosomes rather than being in that chromatin material like it will be for interphase. Each consisting of 2 chromatids connected by a centromere. So when you think about it, you have a chromosome, that's that X shape. Each one of those lines of the X is a chromatid, and then in the middle there's this thing called a centromere, centrocentral. The centrioles migrate to opposite poles. So remember, centrioles are those tube-like structures that are going to help with the splitting of the genetic material. The microtubules start to organize into spindle fibers. This is what's going to connect to the chromosomes. And the nuclear membrane and nucleolus start to dissolve. Because again, mitosis is dividing of the nucleus, and so that nuclear membrane has to break apart for it to be able to be separated. And these are all, we'll talk about this in lab when we kind of are identifying these things, but these things about like the DNA condensing, the nuclear membrane, nucleolus starting to fall apart, starting to have the migrating of the centrioles, those are all visual clues that you can use to tell what phase am I in. Metaphase, this is when the spindle fibers have attached to the center of the chromosomes, and they're aligning on the midway point between the 2 centrioles. So kind of right along the midplate of the cell, you have the chromosomes lining up. So metaphase, middle, and so that's kind of how we always remember when the chromosomes are lined up in the middle, you're in metaphase, mostly. And we say that because these phases can have early and late stages. So you can have early anaphase and late anaphase. And so in early anaphase, the chromatids are just starting to be pulled apart. So, the chromosomes, they're lined up along the midline of the cell, and then each chromatid, because remember, a chromatid is just one line, chromosome is that X structure, and then a chromatid is just one of those, one line from the X. So as anaphase is occurring and the cell is starting to divide, these chromatids are going to start to get pulled apart, and now you have chromatids considered individual chromosomes. And so in the early stage, they're still kind of lined up in the middle, but they're almost slightly being pulled apart. And so you have to kind of understand, that subtlety of, all right, am I in late metaphase, is early anaphase, and we'll get into identifying that in lab. Chromatids get pulled along those spindle fibers, taking them to opposite poles of the cell, so that way they don't get caught up in the cell in the cytokinesis phase, and the chromosome material is being equally divided. And then the last phase is the telophase, the final stage of mitosis. It's when the chromosomes have completed migrations, they're fully on the opposite side of the dividing cell. You start to have the nuclear envelope and the nucleolus reassembling and containing its respective genetic material. The spindle fibers start to disassemble, and then the chromosomes start to unwind again. Rather than being condensed, they're starting to unwind again, so that way they can go back into that chromatin form for functional ability. So here you can see this kind of happening in this cycle. So starting right up here at the very top in this prophase, you can see the centrioles beginning to form. Let's see if I can zoom in. Yeah, there we go. You can see the centrioles beginning to form. You still have, I think I actually called this prophase, and that is not correct, because DNA is still really loose and coiled. It kind of looks like spaghetti in here. It's just kind of all over the place. You can see the nuclear envelope. It has these little gaps, so it's starting to break down, but your nucleolus is still very much formed. As we go into prophase, you can see the nuclear envelope is really gone, the nucleolus is gone, and that chromatin, that loose chromatin, has now condensed into these chromosomes. So again, you have, I'm going to zoom in a little bit more. You have a chromosome, this big X structure. Joined in the middle is the centromere. But each one of these lines, this is a sister chromatid, and this is a sister chromatid. Okay, so each one of these is a chromatid. Together they are made of a chromosome, and in the middle is the centromere. And then over here you have your centrioles. Those are the centrioles. The centrioles send out those spindle fibers, okay, and we'll look at that. So prophase, you have that condensed material, the nucleolus, the nuclear envelope are starting to go, the spindle fibers are starting to form, or rather the centrioles are starting to migrate, they're not just hanging out here together. Then you go into, again, like I said, you can have late prophase, where you see they're still not lined up in the middle yet. They're starting to move there. The spindle fibers are starting to attach to the centromeres. Okay, but then you go into metaphase, where they're lined up along the midplate. They're lined up along the midline, the chromosomes are. We get into anaphase, where those chromatids are being pulled apart. You see the centromere is attached to that spindle fiber, and it's being drawn towards each side. And then in telophase, you can see the nuclear envelope is starting to reform, the spindle fibers are starting to retract. And then as we go into kind of this late telophase, you can see this, what we call this cleavage furrow. This cleavage furrow is forming where you're actually in the cytokinesis, where the cell structures are dividing. Okay, and then you're at the very end, where we're actually back at early interphase, where you have two identical daughter cells. The chromatin has been reformed. Nuclear envelope is coming back. The nucleolus is fully reformed, and the spindle fibers are gone. So all those things are going to kind of be structure or features that can tell you, oh, am I in interphase? Am I in mitosis? What phase of mitosis am I in? And you'll be expected to identify that for our lab. Cytokinesis, we kind of talked about the division of the cytoplasmic material. It begins around anaphase, and then is kind of the defining end of that cell division. It begins by forming that cleavage furrow, which continues throughout telophase and kind of pinches to form the new cells apart. It uses a contractile ring of microfilaments inside of the cell membrane to help divide that cytoplasm evenly. And the two cells might have slightly different amounts of cytoplasm and organelles, but because they share the identical genetic information, they will kind of even back out. As you saw with the endo and exocytosis, cells can take in more water, they can take in more structures as they need to reconstitute themselves. So it's normal for them to be slightly different sizes, slightly different organelles, but eventually, because they have genetic material the same, they can still be functionally equal. And then differentiation. So what can happen after cell division is that sometimes you have cell differentiation, by which a cell develops and specializes into a specific type with specific functions. We have over 290 types of differentiated cells that are specialized for something, and this allows differentiation allows cells to specialize using different parts of the complete genome that is present in each cell. It allows some genes to be turned on while others are turned off. So again, your liver cells are not going to express all of the same proteins that your kidney cells will, but they have the same genetic material. And these specialized cells are specialized. They cannot then become something else. Stem cells retain the ability to divide without specialization. So our bodies have stem cells all over them. Humans, adults of every age have stem cells that act as non-specialized cells. We have certain type of blood cells in our bone marrow that are these stem cells where they can produce one type of cell. They make a cell, and that cell can become 5 different types of blood cells based on the signals that that cell gets. But then you retain these stem cells to be able to produce those other specialized cells. This gives us continuous growth and renewal. The ability of a stem cell to divide and giving rise to at least one other stem cell is called self-renewal. These are progenitor cells. They're daughter cells of stem cells that are partially specialized. So progenitor cells retain the ability to produce cells that will become differentiated in each area of the body. So they're very effective in making sure, again, that our body can continue the process of growth and repair, and it's not just that, oh, well, this one cell is gone now and we're down that cell. That's not how we die. That's not how our bodies start to fall apart. Our bodies keep these stem progenitor cells. Although, after a certain period of time, those might even start to lose their efficacy as well. Here you can kind of see part of that process. You have your sperm and your egg cells. They come together to fertilize the cell. And then this starts with a stem cell. A lot of those first cells that we have after fertilization, because they are what we call pluripotent, they have the ability to become anything, and in fact they do become everything in our body. Those pluripotent cells become everything from your nerve cells and your muscle cells to your taste buds and your hair and everything, and it all comes from these original stem cells. So those stem cells can then produce progenitor cells. Again, they get a little bit more specific, and so your progenitor cells can, this one in particular is making glands and epithelial cells, whereas this one is making nerve tissue cells that help to support nerves function and an actual neuron. So slightly more specificity as you kind of go down the chain versus the pluripotency of fetal stem cells. Cell death is a part of the cell cycle as well. Not all cells are going to go through that divide and differentiation cycle. Some, it is their time to die. It is their time to be broken down. So apoptosis is a form of programmed cell death. It is a normal part of development, rather than something like you get hurt, you get burned, you have a chemical issue or a parasitic issue, something like that. It's not from that. It is very much oh, nope, it's just your time. This cell is no longer developing or dividing properly. So this removes overgrown tissues, damaged cells, and extra cells in fetus, because that absolutely happens, as we're going through, especially that initial stages of development. Our body might produce a little bit too much of a certain type of cell, of an epithelial cell or skin cells, and it will actually start to take those back and kind of scale that back. So some steps of apoptosis: the cell becomes rounded and bulges, the nuclear membrane breaks down, the chromatin condenses, and enzymes cut up that chromosome. The cell shatters into many membrane-bound pieces, and then scavenger cells engulf and affect and engulf and destroy the fragments. So in apoptosis, you are having this shattering, this breaking open of the cell. So for cells that are infected, that are diseased, this is not actually how they are destroyed, because when a cell goes through apoptosis and it kind of breaks apart, that gives you your body or that would give the infected cell a chance to spread that infected material. So we'll see that infected cells are broken down differently. And that's the end of this chapter. Thank you so much.